Dear viewers, greetings. In this video, we are going to see about formulation of immobilized beads-based bioinoculants. The contents of this present video includes an introduction about immobilized beads-based bioinoculants, followed by preparation of alginate bead-based bioinoculants, advantages of immobilized beads-based bioinoculants, and finally, limitations of immobilized beads-based bioinoculants. Immobilized beads-based bioinoculants. Immobilized beads-based bioinoculants are biofertilizer formulations where beneficial microbes are trapped inside the protective beads. They are commonly prepared by using sodium alginate, polyacrylamide or other gel-forming polymers. The process involves mixing microbial culture with polymer solution and dropping into calcium chloride solution to form beads. The immobilized beads act as microbial capsules, protecting the cells while allowing nutrients and metabolites exchange. The immobilized beads provides better protection to microbes against the desiccation, temperature variations and contamination. It also ensures gradual release of microbes into the soil or rhizosphere region and improving the root colonization. And it offers a longer shelf life compared to the traditional carrier-based inoculant, and it is useful in agriculture, bioremediation, and environmental applications such as sustainable microbial delivery. Preparation of alginate beads-based bioinoculants. The preparation of alginate based bioinoculants contains 6 steps. In step 1, the microbial inoculants were grown in respective culture pot. In step 2, 2 grams of sodium alginate was added into 100 ml of culture broth of microbial inoculants and it was mixed for 30 minutes in a magnetic stirrer. In step 3, the mixture was added dropwise through a 10 ml syringe into 100 ml sterile 0.1 normal calcium chloride to obtain uniform alginate beads. In step 4, 1 gram of material contained 16 to 17 beads, each bead approximately weighing 60 mg. In step 5, the beads were washed twice in sterile distilled water and incubated in respective broth containing microbial inoculants for 7 days in an incubator at room temperature to allow microbial inoculants to multiply inside the beads. Finally, in step 6, the immobilized beads were again washed in a sterile distilled water and air dried in laminar airflow chamber under aseptic condition. The alginate beads were then stored in polythene bags at room temperature up to 6 months. This image shows the alginate beads based bioinoculant, that is immobilized beads based bioinoculants. Advantages of immobilized beads inoculants. The first advantage is protection of microbes. Alginate beads provides a protective matrix that shields microorganisms from adverse environmental conditions such as UV radiation, desiccation, and predators. The second advantage is prolonged shelf life. The encapsulated microbes can be stored for several months, up to 6 to 12 months, without significant loss of viability. The third advantage is controlled release. Microbes are slowly released into the soil and ensuring sustained colonization and its activity around the plant roots. The fourth advantage of immobilized beads is high microbial load. Immobilized beads allow high densities of microbial cultures to be maintained. The fifth advantage is reduced contamination. The encapsulation of microbes through the immobilized beads lowers the risk of contamination when compared to free cell inoculants. The sixth advantage is improved field performance. The immobilized beads helps microbes survive longer in harsh soil conditions and enhancing their effectiveness as both biofertilizers and biocontrol agents. The final and seventh advantage is ease of handling and transport. The immobilized solid beads are easier to store, package and transport when compared to the liquid culturals or liquid bioinoculants. Limitations of immobilized beads based inoculants. The first limitation is cost of production. The process, alginate, calcium chloride, sterile condition and equipment is more expensive than preparing conventional carrier-based inoculants like peat and lignite. The second limitation is scalability issues. Large-scale immobilized beads production with uniform size and quantity can be very challenging. The third limitation is moisture sensitivity. The immobilized beads may lose microbial viability if they are exposed to high humidity during its storage. The fourth limitation is nutrient limitation. The encapsulated microbes may face limited nutrient availability inside the bead if not released in time. The fifth limitation of immobilized beads-based inoculant is shorter survival after release. Once the immobilized beads dissolve or release microbes, survival in the soil depends on environmental conditions, that is the temperature and salinity, which may still be unfavorable. The sixth limitation is not suitable for all microbes. Some microorganisms do not survive or multiple well inside the alginate beads. The final and seventh limitation is labor-intensive process. The preparation of immobilized beads-based inoculants requires sterile handling, incubation, washing and drying steps which increase the preparation time. In conclusion, immobilized beads-based bioinoculants offer a major advance in microbial formulation by encapsulating beneficial microbes in a protective polymer matrix. This approach shields microbes from harsh conditions like drought and UV radiations and ensuring more viable cells reach plant roots. The beads enable controlled release, leading to longer lasting colonization and improved benefits such as enriched nutrient uptake and plant growth. Additionally, this system allows for combining multiple microbes in one bead, creating powerful and multi-action inoculants tailored to specific agricultural needs. Despite these advantages, challenges remain in making this technology widely accessible. High production cost, scalability issues, and sensitivity to storage conditions currently limit broader use. Future success depends on developing cheaper biodegradable materials from agricultural waste, optimizing manufacturing, and creating smart beads that respond to environmental cues. With further innovation and farmer education, this promising technology can become a sustainable and effective tool for reducing chemical inputs and advancing eco-friendly agriculture. With viewed through the lens of sustainable agriculture and the circular bioeconomy, immobilized beads technology holds immense promise, and it is a powerful tool that aligns with principle of green chemistry, reducing the reliance of chemical inputs by enabling the effective deployment of biological solutions. By overcoming this current limitation, through interdisciplinary innovation, immobilized beads inoculant are poised to become a cornerstone of efficient, reliable, and eco-friendly crop management system for the future. Dear viewers, thank you for your support. Thank you.